evening, everyone. Firstly, I would like to acknowledge the traditional owners of this land, and I'd also like to pay my respects to elders past and present. I'd like to welcome Professor Stephen Walker, Executive Dean, Science Faculty. I'd like to welcome Terry Irwin, AM, Bindi Irwin, PM, because she's a teenager and always gets up in the afternoon. And that's no disrespect to Terry's Order of Australia. And Robert Irwin. The three of them had just flown in from the Philippines this morning. Robert was there promoting his new documentary, which is all about science. I'd like to welcome the team from Australia Zoo, some familiar faces there. Ladies and gentlemen, boys and girls, this is a pretty special night. If you look around you, I just go, wow, because it's a G20 and we must be the only people left in Brisbane. So thank you very much for coming along here. This event acknowledges two things. One, it acknowledges an alumni of University of Queensland who's been involved in conservation research and has made advances in the field, and of course a very important field given how our planet is changing. And secondly, it acknowledges Steve Irwin. And we all knew Steve from his work on TV, but probably a lot of you didn't realise that there was a lot more than just what he did on TV, but behind the scenes, he was immensely interested in research. And I got to meet Steve, more than a decade ago when I was working up at Lakefield National Park, and I was working on these small freshwater crocodiles, and we happened to bump into each other at a barracks on the national park. And there we spent about 10 days together, and every day we would go off and do our various activities. Steve would be working on animals about 5 times, 6 times the size of my ones, and then we'd come back, and he was, I don't know what the word is, persistent with his questions. And we'd sit down after dinner and he would just ask question after question after question about crocodiles, about evolution, about life on this planet. And if you know anything about science, it's a creative discipline. And to be good at science, to be good at research, you've got to ask informed questions. And that's what Steve did. And he really, really appreciated the work that scientists do. And this is for us, this event merges both science and also the education of the general public. And the motto for Australia Zoo is conservation through exciting education. And that's exactly what they do. Of course, it was a very sad day for me personally, for Terry and the kids, and for the whole of Australia and the world when Steve passed. But Terry and the kids have taken up the challenge, and what they have achieved in the past 8 years is truly amazing. Terry not only runs a zoo with more than 400 staff, she runs 3 conservation properties, a very large property which I just visited just recently out of the Brigalow Veldt. Incredible diversity out there. They're returning this threatened landscape back to its original state. She was just an awardee for an ARC linkage grant. She's a named investigator on that, partner investigator. As we know, Australian Research Council grants are difficult to get. Australia Zoo has 3 of them in a trot for the research, and I think Hamish will be talking about part of the research. So that really deserves an acknowledgement, Terry, for your contribution. Part of that is contributing not only the incredible team at Australia Zoo, but an enormous financial contribution to support the research. And again, it just continues on the tradition that Steve, I know, would have supported 100%. So tonight we've got an exciting presentation ahead of us, and I'd like to invite Professor Stephen Walker to come to the stage and to introduce our speaker. Thank you very much.
Thanks, Craig. I'd also like to acknowledge the traditional owners of the land where we gather, and where in the next few days the whole world will gather. The traditional owners were here for many thousands of years and looked after that land, and I pay them my respects. I'd also like to welcome once again Terry and Bindi and Robert and all the staff and all the great friends and colleagues from Australia Zoo. It's a real pleasure to have you here again at this annual event to commemorate the work of Steve Irwin and the wonderful influence that he's had on an entire world of conservation and biodiversity. Each year when we hold this event, it's my pleasure to introduce the speaker for the evening. Now, 5 years ago when we first held it, the speaker was Craig. And in the intervening years, we've heard about frogs, we've heard about crocodiles, we've heard about Komodo dragons. Tonight we're going to hear, I think, about a whole range of animals, fishes, other things. And it's my very great pleasure to introduce Hamish Campbell. Hamish runs the Movement and Landscape Ecology Laboratory at the University of New England in Armidale in northern New South Wales. But Hamish has actually worked and studied all over the world and has had the opportunity as a result to study animal movement, animal behaviour, physiology and so on in a range of different settings. He did his PhD at Plymouth University in the UK. He spent 4 years in the British Antarctic Survey doing his postdoc fellowship, including quite a considerable period in Antarctica. And somehow Hamish was telling me just earlier outside, he got waylaid on the way back from Antarctica and ended up in Brazil, where he spent several years in a somewhat warmer climate at the State University of Sao Paulo. Then he came to UQ and spent a number of years working with friends and colleagues here at UQ, and it was wonderful just earlier to see so many people greeting Hamish again and welcoming him back to UQ. And now he's at UNE doing this marvellous work about animal movement, animal tracking. And so with great pleasure and no further ado, I'll ask Hamish to come and tell us about his marvellous work. Welcome, Hamish.
Okay, let me just... Does everyone hear me okay? Is that good? Okay, good. I've got to be careful not to walk in front of this. Animal movement reflects and influences the distribution of ecological resources in space and time. Okay? And therefore, to conserve and protect ecological resources, we must have a fundamental understanding of animal movement. Now, we're not all ecologists here, so let me just put that in a different way. Animals move through air or the water or across the landscape primarily to look for resources and to find mates. Okay? And whilst during this action and these movements, they'll spread pollen, move around seeds, inorganic nutrients, energy, and food sources, even animals. Okay? Now, I like to think of animal movement really as like the glue that really ties the ecosystems together. Okay? It's a good way to look at it. It's like the ties that bound the ecosystem. And we're beginning to understand now that we can't conserve animals in isolation, okay? We need to conserve ecosystems in order to conserve animals and conserve wildlife. So therefore, by unraveling those ties and looking at them individually, at those movements of those animals, we really have a better understanding of the whole, okay? Now, I was 40 this year, okay? I know I don't look it. I am from Scotland, you probably noticed, and this is what happens when you don't grow up under the Queensland sun. Okay? But I was 40 this year, and I've really spent it made me realize that I've spent the last 20 years chasing animals around the globe, gone to some amazing places, really to try and understand not just where they go, but why they go there, who they go there with, and what they do once they get there. And what I want to talk about tonight is really try and give you appreciation of this field of science, because it's really quite an exciting field. Really, so when you go out there and watch a TV documentary, and the next time you're seeing some of the scientists screwing a device on animals, because it's really common these days, you'll begin to have a better appreciation on why they're doing it, and indeed tell the good ones from the bad, because there is some bad ones. But where did I start? So I want to go back to the start. So did I get this appreciation or this love for animal tracking and animal movements? Well, when I was younger, I really had an appreciation for wild things and discovering secret places. And I think all children do. Hence the success of Morris Sedgwick's book, which I've taken the title or adapted it slightly for this talk. I'm sure many of you have read it as a kid. But when I looked at actual animal tracking, I had to think about it when I was putting this talk together, and I thought, well, where did I get animal tracking from? And I think I can actually trace it back to a single event when I was 8 years old. Okay, so this is me at 8 years old. Thanks. In usual Scottish attire of summer attire of shorts, t-shirt and wellies, or gumboots as you call it over here. But it was funny, I had to, because we live in a digital age, I actually don't have any photographs of myself before digital cameras on film, so I had to phone my mum up over in Scotland and ask her and say, Mum, can you send me some pictures for this talk? And she sent me this one, so I haven't actually seen this for 30 years. And if you look, it actually says Save Wildlife on my t-shirt. Now, that's pretty radical for an 8-year-old in 1982 Scotland, believe me. But anyway, I want to draw your attention to the picture on the left there. So this is Henry. This was our tortoise. This is a Testudi hermanni. So this is the common sort of southern European tortoise. Now, I don't know why, but when I was growing up in the early 80s there, we had this TV program called Blue Peter. I don't know if you got it in Australia, but they actively encouraged children to keep tortoises, these specific tortoises. So thousands of these tortoises were shipped from their native southern Spain to pretty much live a Scottish winter. And we used to have to put them in boxes to hibernate and stuff. But anyway, Henry was a real explorer in our garden, and he used to go everywhere under my mum's rose bushes, climb under the fence, into the next door neighbour's garden, and into the next. So what I did is I actually painted a big letter H on his back using my mum's red nail varnish. And we used to be able to track him down, but it didn't work very well. He still used to get lost. So one day, actually, I watched this TV documentary. I'm not sure what channel it was on, or we only had 4 terrestrial channels back in those days. But I watched this documentary, and it was about these guys out in Yellowstone, and they were putting radio collars on bears to figure out where they go. And I just thought, that's the ticket for me, I want one of them. So I said to my mum, can we get a radio tag so we can tag the tortoise, we can tag Henry, and we can track him and figure out where he's going. So Christmas morning arrives, come running down the stairs, big parcel, rip it open, microscope. My mum thought, love of science, get him a microscope. I still hate microbiology to this day, I tell you. Now, I know the little things make up the world, but if it's not got a backbone, I'm just not interested in studying it. So anyway, we didn't get the radio tag, and eventually we did lose old Henry. Never found him again. I blamed my mum incessantly. When you're 8, everything's your mum's fault, of course. Never found Henry, and I believe really that set the seed that then after that I wanted to tag animals and got this love for understanding where animals are going and to try and find them. That's where it comes from anyway. And actually my mum came over a few years ago, and this is the truth. She came over a few years ago, and we spoke about old Henry when I was looking for these photographs, and she said, actually, we never told you, but old Henry wandered onto the drive and got run over by the postman. The post truck, yeah. No open coffin. But that was it. So anyway, and so all these years, life could have been so much different. So there you go, never lie to your children. Anyway. Let me just get some water. So, I want to go on. So, I did tell you there that I watched that TV documentary. And when I was putting this talk together, I managed to actually track down, sorry, I'm going the wrong way, a picture of those original guys from that documentary. So these are the Craighead brothers. They were really the pioneers of radio telemetry back in the 60s. And they were the first guys to radio collar bears in Yellowstone. John and Frank, I believe Frank's still alive. They're probably in their 90s now. And what they're doing here, you can see this is a nice brown bear, and they're putting a radio collar on it. Now, for those of you who don't know how radio tracking works, is that the radio collar goes on the animal, as you'd imagine, and it sets up a pulse at a set frequency, like a radio frequency. And each animal's got a different frequency. And that's just radiates out from the animal as he moves around in the landscape. And then as you can see in Frank's hand here, he's got the receiver. And this is a receiver aerial, and how it works, it's kind of directional. So when the aerial's pointing towards the animal, the signal's much louder, and as you move it away, it gets quieter. And then using that method, you can go in the landscape and track animals and sort of triangulate and get their position. And that's really what they're doing there from back in the 60s. And with the wonders of the internet, I managed to actually track down some original video footage of these guys doing it that I'm going to show you just now. It is a little bit grainy, so I do apologize, but it was 1967. But when you watch it, you'll begin to really realize how these people back then, almost 40 years ago, how they're really facing some of those, challenges that we face today in animal tracking. So, without further ado,
try that. I don't know. I'm trying, but let's try it. See. Let's get one in. Okay. Watch this.
Okay, so maybe a few things have changed, okay? A bit better OH&S, a bit more professional. We get you to these. We don't use our own vehicles for starters. That's a lesson there. So anyway, so that was back in 1967, and then pretty much things stayed like that for a long time, throughout the 60s and into the 70s, and almost for 2 decades, people just tracked animals using exactly the same technique pioneered by the Craighead brothers. But I want you to draw, I keep pointing this up there where it's here, draw your attention to this slide. So this is just something that happened. This is by 1973, the US launched, the military launched observation satellites. Okay? And the reason they launched them was to track vehicles and tanks and soldiers and stuff. And they were tagging them, so what they'd actually do is they'd have like a remote device on the vehicle, and they'd track them with these observation satellites. And they don't work like your standard GPS that's on your phone or on your sat-nav in your car. This uses something called the Doppler effect. So if you can imagine, the Doppler effect is like when an ambulance goes past you when you're in the street, and it gets louder and louder, of course, as it gets closer towards you, but the pitch also goes up, and it actually gets higher. And then as you go away, it gets lower again. The pitch gets lower. And that's exactly the same effect that these satellites, as they pass overhead, and you've got to imagine there's a beacon on the ground. As they get closer, the signals get higher in the frequency, and they detect those higher frequency bands, and then they get lower. And that's how they started doing this back in the 70s to track military personnel. Okay? Oh, so that's important to know. As we go on our next slide, but it wasn't for another 10 years. It was in 1983, so this is almost 20 years later. the Johns Hopkins University, their applied physics lab, a guy called Tom Sidwerka, he tracked the first movements of an animal using a satellite. Okay? And this isn't what he tracked. It wasn't a bear or a moose, it was a mute swan. This isn't actually the mute swan that he tracked, but it looks very much like it. And I did try and actually find some data from this program, but it was very difficult. So 1983, point to remember, first ever animals tracked by satellite in the world. Let's move forward 3 years, and we've got this motley crew here. Some of you might recognize some of these faces, but this is, does this have got a little red thing in it somewhere? Okay. So some of you recognize, this is Lynn Beard, who's a technician. Is Lynn here? No, she's not. So this is Lynn Beard, and this is Professor Gordon Gregg, who was a professor here at UQ for some time. But this is they actually did this at the University of Sydney, and they were the first ever people to track an animal by satellite in Australia. And the animal that it was one of these. It was a camel. And this is some of the original photographs donated to me by Gordon of some of that early pioneer work when they're satellite tagging those camels. And that's the tag going on it up the top here. This is the tag. And I really like this photograph. This is amazing. So these are the two satellite tags, and then this is the 1986 skyline of Sydney, where they put the tags on the roof. And you can see they're really quite clumsy sort of things. When you see, I'll show you some footage from what we're doing today in Australia, and you'll see that we've come quite a long way. But anyway, this is in 1986, and the slide I put in next, this is a quote from Gordon in one of these papers that he wrote back in 1986. And I'll just let you read it there while I have a drink of water. Okay, so the reason why I wanted to put this up was that even back then, so back in the 80s, the scientists were beginning to realize that technology was going to unlock, unlock some of those secrets that we had about nature. And we were going to be able to start looking at how animals move in landscapes and manage them better through that. And really give up their secrets. So that's back in 1986. What Gordon was wrong about really was that it wasn't actually, the time wasn't close. So back in 1986, nothing really happened again for a number of decades. So what happened? The 90s came and went. Okay? Now, that was pretty cool back in 1991 in Scotland. Okay? That was very cool. Look at Noel nodding his head. That was pretty cool. But anyway, the 90s came and went. And I put this slide up not just to show you my great fashion sense, but I just wanted to show you is that this is really where I joined the story, okay? So I graduated in 1996 from Dundee University, and this is really where I started getting involved in tracking. And we're going to get rid of that photo now, before Bindi wets herself. Sorry. You can have it. So anyway, they were hair extensions, by the way. They weren't my real hair. Even worse, weren't they? Don't know what I was doing. I thought it was cool. But anyway, so 1996, I graduated. And we're going to just diverge from satellite telemetry for a moment. So keep your mind, remember about all the stuff I've talked about satellite telemetry, because we'll come back to it in the talk. But we're going to talk about, in the 90s, something else came along called passive underwater acoustics. Now what that is, it's basically similar to your radio tracking, but underwater. You put the tags, I've actually got one. So this is one of these tags. We implant them inside the animal, and they send out a sonic pulse, and we detect it from putting stations on the seafloor, okay? Now, the reason why we're using this and not satellites is because you can't track marine animals underwater, of course. Satellites are only good for the air. So how do we look at animals that don't surface? And what we started using was acoustic telemetry. So back in 97, I went out. This was at the University of Hawaii. I was part of this program under Dr. Kim Holland. And what was happening was that tiger sharks, there was a spate of shark attacks around Oahu on some of the tourist beaches. And tourism's big for Hawaii, so people getting eaten by sharks isn't good for tourism. So they used to go out and they'd send out these big culls, drum lines, nets, similar strategies, similar story. that we can see with white sharks today. And they were going out and killing these animals indiscriminately. Now, what Kim Holland did is he lobbied the government and said, look, there's this new technology out called underwater acoustics. When we could track these animals, we can implant these tags, and then we can put these listening stations on the tourist beaches and get an idea of what sharks are coming in to the beaches and watch their behavior and try and understand their movements. So that's what they did. They tagged those sharks, and what they actually found was that these animals were completely nomadic. They didn't show any home range behavior at all. So if there was an attack, the chance of you went out the next day and culled a load of sharks, the chances of actually culling the culprit, the shark that made the attack, was very slim. And thus, culling of sharks stopped in Hawaii on the basis of this data. So I just want to tell you that story because it really shows how by understanding the movements of the animals, we can really better manage them. It's really important. Now, in Australia now, with white sharks, we're doing exactly the same thing. So almost 20 years later, we're tagging white sharks with exactly the same technology as we were doing back in the 90s. Unfortunately, we're not sure if they're going to listen to the advice of the Western Australian government, and they're actually culling the sharks. Anyway, after the 90s, Stephen said I went out and worked for the British Antarctic Survey, and we actually took that same technology and we took it down to the Antarctic, and we started looking at fish migration around the Antarctic. Well, of course, Antarctic fish don't attack and kill people, but the purpose of that was to look at fisheries, so it was to create fisheries policy and to create better fisheries networks. And again, this technology now being used routine all around Australia to look at fish stocks and better, give permits to who should be fishing there. I just thought I looked kind of a hero in this photo here, and I just tried to crowbar it into the talk somewhere. But anyway, so this is actually me coming through an ice hole. This is an ice hole in the background here, and the picture's taken from underneath. And we're coming down to service one of these underwater stations. So I just want to jump. So around about 2000, this is going back to satellite telemetry. So different type of telemetry. But something really amazing happened in 2000. Really monumental and really changed. It was a game changer for satellite and animal telemetry. And that was the US removed something called selective availability. It was a forced error. A lot of people don't know this, but GPS satellites were been around since the late 70s, early 80s. The global positioning satellite system, that is. But what the US military did is they put a deliberate error of about 100 meters into the locations that you got from the satellites. So only they could get really good data, and the rest of us were all incorrect. And then what they did in 2000, they removed that, and hence we see this large explosion in the use of GPS devices and sat-navs in your car, in your phones, and handheld devices. But that's why they weren't used before. The only people that really used GPS before 2000 were sailors on ships and stuff like that. So GPS, I talked earlier about the other satellite system, that Doppler effect. So GPS works very different. It triangulates. So how your GPS works on your phone is, or in your car, just like the photograph here, it's the active unit, and it looks for these signals coming from different satellites. And then based on where those signals are and the distance they are from the satellite, it calculates your position. And it's very accurate. Your sat-nav's accurate probably to a couple of meters. So that happened in 2000. And then something else happened. It was the rise of mobile technology. So mobile phones, again, for us scientists in animal telemetry, really were a game changer. It wasn't just the technology. So your smartphone nowadays, I heard something the other day. They said that a mobile phone's got 200,000 times the computing power of the Apollo missions to the moon, which is quite amazing. So the reason why they've got a lot of money. Mobile phone companies got a lot of money to spend on research and development. So not only did they advance the technology and the chips within the phones, but they made them smaller. They reduced them and they made them miniature so they'd fit in your pocket or your handbag. And what they also did is they reduced the power consumption. So the chips your phone and your chips now, compared to 10 years ago, are really, really power efficient. And then the other thing they did is they brought down the cost because they wanted to make it affordable to everybody. So those three things, cost, power, and miniaturization, enabled us biologists to package up those units and stick them on animals. And that's indeed what we've been doing now. And what we've seen is an explosion in the use of animal telemetry throughout the world. Now, I Googled animal telemetry before I came, and this is what you get. You Google animal telemetry and look at images, you just get these hundreds of pictures of people. You would think that the only reason why we tag animals is so we can get our pictures taken with them. Thousands of these photos on the web. But the reason why I put this slide up, though, is to show you the diversity. Look, we've got crabs, we've got turtles, we've got penguins, we've got seals, we've got birds, we've got crocodiles, we've got bears, sharks, everything. So animals everywhere are being tagged. Literally thousands of animals worldwide. Probably in Australia there's thousands of animals at the moment with active devices on them, monitoring their movements in really quite high frequency and high level of accuracy. And our problem really is what do we do with that data? Yeah? And that's really where we are at now in the field of movement ecology. And this is why we call it an emerging paradigm, because we've suddenly enabled ourselves with a huge amount of technology, and we're collecting these huge amounts of data, but we're not really sure what we're going to do with them all. And I kind of liken it to like when Galileo first created a telescope and looked at the stars, or when Robert Hooke created the microscope and was the first person to look at cells. So that's really where we are in the science, where the technological innovation is really leading a conceptual advancement in our science. But we need the statistical tools to do that, and that's really why it's such an exciting field. And we're still really trying to figure out what all this data means. Big data, we call it. So what I'd like to do now is just show you a few examples on some of my projects here in Australia to show you how we're applying some of that movement data to management issues in animals. Okay, so this slide I think was somewhere at the start of the talk. This is an estuarine crocodile taken up, I think he's called Drew, this crocodile. He was caught up on the Wenlock, and this is a satellite tag you can see on the nickel shield on its back here. Now, estuarine crocodiles, incredibly controversial and political animals, okay, in Australia. I don't know if you saw, just on Tuesday night, they caught a 3.8 metre crocodile in the Mary River. It's almost eclipsed the G20 in terms of media coverage. You know, it's like incredibly political animals. If you go up to the top end, everybody's got an idea on how crocodiles should be managed. Everyone's got an opinion on what we should do with the crocodiles. So this is my opinion. But what I believe, though, is that no matter what you, and this is what I say to a lot of the guys that work with the governments, is it doesn't matter what you think, the crocodile issues are and what you should do with them. Australia should be very proud of their estuarine crocodile population, because we live in a world of real ecological gloom, really, where a lot of the top predators across the world are declining. And here, the estuarine crocodile was brought back from almost extinction. We estimate that in the early 70s, there might have been as few as 3,000 crocodiles. So they were on the edge, on their way out, and through management programs and bans and the ban of hunting, that population has come back now to a really healthy, substantial population across the top end. Now, crocodiles are dangerous, and they do occasionally attack and kill people and livestock and pets. People hate it when their pets get killed. But because of that, whenever an attack happens, I always read in the papers and across the internet all these calls for culling. We usually call for, let's go and cull the crocodiles. Now, when you sit down and think about that, and this is what I tell people, is that that's a really ridiculous argument to go out and cull them, because what level of culling do you consider be acceptable before you go back and swim and say it's safe? Should we cull 25% of them, or should we cull 50% of them? Really, only 100%. If culling is going to reduce attacks and that's your purpose, then surely only 100% is the way to go. But we're not going to do that, because we've come a long way since the 70s. So it's a ridiculous argument, because we're not going to go back there to that point we were back 40 years ago. So what we've been doing with Craig Franklin here at UQ and Terry with Australia Zoo and all the other guys here is we've been involved in a monitoring program to look at the movements of estuarine crocodiles to better understand their social behavior, look at how management actions may impact upon them, and then look at how they actually behave around sites of human habitation. And what I'm going to show you now is I'm going to show you a video that really just explains it all. It was put together by Luke Burnett, who's a media officer with UQ. This is a short video that was filmed in 2013, and it's really quite good. And it really shows you quite nicely some of the research we're doing, but it also shows some of those technologies we talked about. So you'll see acoustic tags going in the animals, and you'll see satellite tags going on the animals. So. Pretty cool, eh? But I don't really need to say any more about that. We're just going to, that's the crocodile research program. Anyway, on to turtles. Something far cuter. Okay? So, this is the penny turtle. Is Marilyn here? She is. Hey, Marilyn. Marilyn's photograph. But isn't that cute? So the penny turtle is the Mary River turtle. Okay? Elusa macrurus. Now, I want to tell you about this because it's a great story, but also it really as well exemplifies or demonstrates why understanding animal movement, or sorry, not understanding animal movement, but not having sufficient information about animals or where they exist can really lead to their demise. Okay? So the Mary River turtle, very cute, only lives on the Mary River in Queensland, and which is only 3 hours north of Brisbane. But in the 60s, if you had a turtle in the 60s or 70s in Australia, it was a good chance it was one of these. It was a Mary River turtle. Now these were called the pet shop turtle or the penny turtle. And as you can see, they're incredibly cute. But the other thing that the Mary River turtle has is they hatch just before Christmas. So they were just perfect for the sale in the pet shop industry. And a fisherman from up in Maryborough, he realized that these turtles, after rain, you can see the tracks where the turtles come out of the river, and you can see where the nests are. And if you dig down, you can access the eggs. And what this guy was doing is he was going up to Mary River, and he was collecting these eggs. And they estimate that he was collecting around about 15,000 eggs a year. Now, because he wasn't sure about how to keep those eggs, or he wasn't a biologist, a lot of them died. So he estimated, he was still alive, and a master's student a few years ago from here at UQ interviewed him, and that's how I got this information. But probably about 25% of them died, and they never hatched. Now, it's easy to sort of think this is terrible, in our modern day. Think, oh, how terrible was this guy going out and taking these animals. But he didn't really know what he was doing. He didn't know that this turtle was only found in the Mary River. They were actually selling it in the pet trade as a different turtle called Elsea latisternum. And it was a turtle enthusiast from down in Sydney called John Cann who recognized that this little turtle that they were selling wasn't in fact the Mary River turtle. wasn't in fact a losing wasn't in fact Alcela I'll get it right. It was something new to science. It wasn't even a closely related species. It was something completely new. But the pet trade's got a bit of a code of secrets, and it won't let up. You know, we won't tell them where their suppliers are from. So John never actually found. He went to the pet shops. They wouldn't tell him where they were getting these little turtles from. So he called it short-necked alpha. And for the next 25 years, John searched for the habitat of short-necked alpha. And he dived all over Australia in hundreds of river systems. He even dived in the Mary River. But if you've ever been there, it's really murky, and no wonder he didn't find it. He went to Torres Straits. He went up to PNG. He went all over looking for the location of short-necked alpha. In the 80s, you know, there was a ban on the selling of native wildlife. And eventually, John managed to get the information he was looking for, that these were coming from this guy from up in Maryba. And he went and visited him, and they took him out to the river, and he found the location and the habitat of the Mary River turtle. And they formally described it in 1994. Now, I think that's quite remarkable, because this is a turtle, they grow to about this big. They're only 3 hours north of Brisbane. The highway even goes across the river. And nobody recognized that this population was living there. When they found it and they discovered, and they renamed the population, they estimate it's only about 5% of what it originally was back in the 60s. Okay? So even though we just discovered it, it looked like it was already on the way out. Now, a community group based up there in Tyro, called Tyro District Land Care Group, they took on board to try and protect this turtle under the leadership of Marilyn Connell. And what they did is they sold chocolate turtles to raise money to try and protect the nests, the few nests that were left, as well as invest in research and science projects here. And the next slide, this is my wife, Mariana. She actually got one of those scholarships, along with an IPRS, and undertook a study on the Mary River, looking at the movements of these turtles in the river system. And this photograph here, as well as a really nice picture of my wife, shows, you can see the tag there on the turtle. And you can see it's tiny. So the technology's come, you know, so advanced now that we can put these really small little tags on really small little animals. And what we did is we tagged these animals and released them into the river. Let me just step back a bit. We couldn't find the hatchlings. So we didn't actually know whether the nest protections were doing any good. So what we actually had to do is we took the eggs from the wild, and then we hatched them out into turtles, and then tagged them, and then released those ones. And after a while of tracking, using that same technology that I talked about, the tiger sharks, we were able to find that these turtles were actually being located in a very narrow range of habitats within the river. And what those habitats were these sort of rocky sections. They call them riffle zones. They're sort of shallow areas within the river. They're very characteristic of the Mary River. They're sort of these pool riffle sections. And the turtles were all moving to the tops of these areas. Now, this is important because if you think of damming of a river system, you're going to raise the water level, and bang goes your riffle sections. So damming of the river there would be very detrimental to the Mary River turtle. Okay? So that's just a nice little story there on how, by understanding the movements of those animals, we're able to inform policy. And that information is going into the recovery plan, and hopefully we'll be able to protect the river from further impoundments. Okay, the last little story I want to talk about is the southern cassowary. So the southern cassowary, I'm sure you all know what a cassowary is. You know, until I arrived in Australia, I actually didn't know what a cassowary was. I'd never heard of one before. I also thought there was only one species of kangaroo and one wallaby. How about that for a biologist? But I actually think if you go to the UK, most people will only ever think there's one species of kangaroo. Anyway, the southern cassowary, a magnificent animal, prehistoric. If anybody's gone up to the wet tropics, you can go down to Mission Beach, or there's a little place called Etty Bay. You're going to see these things in the wild. Magnificent creatures to see. And the cassowaries are so prehistoric that if you took the phylogenetic tree of birds, you would see that at the bottom it branches. And on this side, on this branch, we'd have cassowaries, rheas, ostriches, what we call the ratites. And then on the other branch, you'd have all the other birds. That's how ancient they are. From Gondwanaland, the original Gondwanaland animal, and they're flightless, so they walked all the way to the wet tropics. And prized by Aboriginal cultures. In PNG, they use them as a dowry for when they marry off their daughters. But prized bird, I'm not sure how they taste. But, what should we say? Yeah, prized bird in the system, but if anybody goes to the wet tropics, you'll see that there's not much wet tropics left. Yeah? If you go in the wet tropics in Australia, you would think that rainforest only grows on mountains. Why is there no forest in the valleys? That's because they chopped it all down for mangoes and sugarcane. So the cassowaries, from having this contiguous habitat, are now living in these little islands in a sea of modified habitat. They live on these little island peaks of rainforest, and when they get saturated or they have little chicks, to get to the next piece of habitat, those animals then have to cross farms, roads, urban areas, and this is where they get smashed by dogs and cars and traffic. Not particularly good at crossing roads, cassowaries, a big mortality factor for the birds. As you can see, they're black. People don't see them. So, a few years ago, I was up there doing another project, actually, when I saw the cassowaries and thought, yeah, these are amazing animals. I want to tag one of them. I'm not done with birds yet. So as you can see, this is a satellite tag on the bird here, on its leg. And they said it could never be done. This is the first ever ratite that was satellite tagged, that we tagged up here. And what we were interested in was, because the birds were living within these mountaintop zones, the Queensland government had said, we're going to protect these areas, and we're going to make these cassowary conservation areas. Really, that was because they're not useful for anything else. They're just these mountain, no useful for farming, so let's conserve them for cassowaries. And the question I asked was, well, does these little, conservation areas, does they encompass the full movements of the birds? So that's what we did. We went in there and we caught all the birds and we tagged them all with these satellite devices and monitored just to see if they actually stayed inside the reserve, or were they going outside the reserve where there was dangers, where they were threatened. And what this little video I've got here shows, so this is the reserve. So this is called Mooresby National Park. It's along a river system here in the wet tropics. And as you can see, completely cleared. All that forest gone. Because when the early settlers came to Australia, they actually gave them the land for free. And if they didn't clear it, they had to give it back. So they completely cleared all these areas, and the cassowaries are now stuck in these little islands. And what you can see here, this is a home range of a cassowary. So a home range is like, we look at a number of locations from the tags the animal's wearing, we do a couple of algorithms, and we create this home range area. And the home range tends to encompass the daily activities of the animal, where it looks for resources and shelter. And this little, this is one of the birds here, this little 667. And if I just click it, we can see some of the movements of the birds. And what you can see right away is the birds leaving the park. This is the national park. And the birds are going over here, forage, and then come back to the park. So the park is really good at encompassing the most, 90% of the movements of these birds. But all our birds were doing this. They were all going over to this little patch over here when we looked at the data. So we thought, well, what's going on? Where are they going? What's so exciting across here? Because this little area over here, this is almost a kilometre that those birds have to walk across paddock to get there. So we went over there to investigate, and what we found was this thing, pond apple. So pond apple, Annona glabra, this is it here, this is the fruit, beautiful fruit. Cassowaries love pond apple. They love bananas and mangoes as well. They actually prefer it over their native fruits, higher sugar content, I believe. But the birds were going out and feeding on pond apple. Now, pond apple is an invasive weed from South America. It's a class 2 weed under the Queensland government. And they've had huge eradication schemes in the 70s and 80s to get rid of this thing. It's incredibly resilient. So it was brought over, and what they did is they brought it over and they tried to graft it onto custard apple. So they used the pond apple because it was really tough and resilient to grow custard apple. And what happened? It was more resilient than they thought. This thing escaped and went, well, classic story, classic Australian story, actually. But This thing escaped and started coming. It can grow in mangroves. It'll take over the mangroves. It can put its roots down in fresh water. It can grow in the rainforest. It's an unbelievable plant. If it wasn't such a noxious weed, we'd think it was great. So what the cassowaries do, and we're going out of here, and what we found is we looked at these cassowary scats. We found these cassowary scats, and this is completely made up of pond apple seeds. So the cassowaries were definitely going out of the reserves, feeding on pond apple, and then they were going back to the reserve, and this is a little stand of pond apple growing out of a cassowary scat in the rainforest. So these birds were going out, so picking up those seeds, taking them back to the rainforest, depositing them in a really nice fertile dollop, and they were almost causing 100% germination of the seedling plants. So this is just an ongoing, this was just really an observation, and we're now working with some other colleagues at CSIRO to look at sort of the seed dispersal models, to look at that interaction between the invasive animals and the cassowaries. Because cassowaries, we all think, and it's often proclaimed, they say they're the gardener of the Australian rainforest. Because they replace mammals in other rainforests. You know, they're monkeys dispersing large fruiting plants. Whereas we don't have it, so it's the cassowaries. But they're also incredibly good weed dispersal agents. So this was the first time we observed this, and we're still looking into it. But that's just another aspect why looking at movement ecology can really begin to help us to understand how we can better manage not just the wildlife or the animals, but the resources they feed upon. So that's really, I'm coming to the end of the talk now. I think I'm pretty close to out of time. But that's really just a few examples of some of the work that we're doing down on our lab, or have been doing for a number of years. Just to finish up, I think we want to go back to this slide again that I showed earlier, just to really show, I really just want to hammer home that point that movement ecology is an expanding science. We've got a huge amount of people are working on it and tagging animals out there. And you will see that there's not necessarily the data's, what can I say? What's happening is a lot of people are tagging those animals, and then that information is not being made available to the scientific community. And it is a big problem. It's something that I've been working on for the last few years. We've been trying to encourage to create much more collaboration across the community, because what's happening is people are going out and tagging animals, and then NGOs are tagging animals, the government are tagging animals, researchers are tagging animals, and then that data is not being used for anything. It's not actually making it in the scientific literature, or if it is, the data is not being made available. So I think it's a really important point, and those PhDs, they do their projects and then they leave and they take the data with them. It's on their hard drive, but it's no use on their hard drive. You know, the tagging is an expensive science. It's really expensive to tag animals, and there's also an ethical concern, because we're actually handling those animals, we're causing a level of stress to them by handling them, and we're attaching devices to them. So I believe that we have an ethical consideration to make the most of that data. And one thing we're working with is trying to make that data open access within Australia. So that's a little bit of future. I just want to finish up. This is the last slide. And I'm going to show you another little video. This is a great little video because it sort of shows you the future, what we can see into the future. And how by looking at animal movements and overlaying them with environmental information, we can really get a good idea of how the animal, lives within its environment and how it uses resources. So if we're going to protect those resources, we can do it more efficiently. And protecting mobile species is particularly difficult because they're, say, moving around the planet, they're going to different countries, there's different legislation. How do we, collaborate across national boundaries? So this isn't my work. This is done by the University of Amsterdam. And what it is it's the tracking of 2 honey buzzards. The blue is the male and the pink's the female. And what the tags have an altitude. So what you see here is almost in 3 dimensions. You see longitude and latitude, so the 2 dimensions of space, and then that 3rd dimension of height. Okay, so what we're seeing here, this is the male. So you can see his home range. He's obviously got a nest there in the blue circle, and he's out there feeding. And then the pink's the female, and you can see she flies a lot higher than him and ranges a lot further. So and now they take their winter migration. They're moving through Europe. They're going through the Straits of Gibraltar, Morocco, and the male comes down here to Senegal, and the female continues all the way down to, I think that's the Republic of the Congo. They make the return journey in spring. The female actually goes a little bit off course. And this is where we join her again as she heads off through the desert. And you can see she is making stops in the desert there, on the ground. She seems to be a little bit lost, I think. What she's feeding on, or we have no idea. I mean, the migratory potential of those animals is quite amazing. But they come back up the way they came. They go back through Spain, the Straits of Gibraltar, straight back to where they were. So the male arrives first in the season, creates the nest, and here comes the female to nest alongside him. Why I think that's quite amazing is because you just saw the entire annual cycle of a pair of buzzards. And in 1 minute, within 60 seconds, you not only did you see that, you understood exactly where those birds go and what they do. So the power, you can see the power of movement ecology and visualizations when we begin to put them together. It's really quite sexy science, but it's also a really good way to communicating science to other people. And we go those movements. I just want to finish, like every talk, with some acknowledgements. I need to thank Craig Franklin and Terry Irwin for inviting me to give this talk, UQ and the Australia Zoo for hosting it, all the colleagues and collaborators I've been with throughout the years, and without a lot of this work wouldn't be there. Everybody in Australia, I'm now a resident, way. We're going to get our citizenship this year. Australia's a great place, we love it, myself and my wife. And really for sharing all these wild things and wild places that you have in Australia. You know, there's so much yet to find out. You know, I showed you the example of the Mayor River turtle, only described in '94. We're also working on a shark called the spear-toothed shark up on near the Steve Irwin Wildlife Reserve, and we've never caught an adult. We don't know what the adult sharks even look like or where they go. How amazing is that? So what we're doing is we're catching the juveniles and tagging them. And that's the photo there is from the Mayor River. That's myself and Mariana. But thanks a lot. You've been a really good audience, and I really need to go get a beer. Thanks. But I think we're on to questions.
Thank you, Hamish. There's time for a few questions, if anybody's got any questions.
Okay. Well, I guess there's 2 points there. The first is that technology is actually available. I think there's some companies making, not tracking devices like the GPS, but like an RFID, we call it. So it's like a tracking device. So you could go and find your cat if it got lost. In terms of actually tracking their movements individually, I guess the problem with that is we'd all come back to like animal ethics issues. You know, we've got to go through a lot of ethics to, even I actually worked on a project, we were losing domestic dogs and we had to go through a lot of ethics. So the ethical consideration of giving that for people to use, I'm not really sure if that would be possible. But there was a BBC documentary, I don't know if anyone saw it, it's on YouTube, where they tagged a whole load of cats in this village in England. They tagged all the cats and they looked at how they behaved and who visited who and who's raided who else's food and stuff. So the technology's there, but I don't know if it's commercially available or not.
Are there any, Terry?
What do you think the next
Can you
step
repeat the
is
question?
with tracking? So when will our tracking devices get smaller but still have extended battery life, which every mobile phone user is wondering as well?
Okay, so Terry was asking, when's the technology going to get better? When are we going to get even smaller and more power out of our tags? The thing is about the tags is we're always related to the power. So even though we're evolving in chip technology and stuff, batteries have pretty much stayed the same for decades. So until we manage to get over that, of some way of, you know, storing power in a miniaturized device, we're always restricted by power. So something that the Max Planck Institute, actually the NASA are involved in, and the European Space Agency, and we're not involved in Australia yet, but we're trying, but it's called Icarus. Now, Icarus stands for, I can't remember, something about, anyway, they crowbarred this acronym in Icarus. It's something about tracking animals using space. But anyway, Icarus is, they're actually putting antennae on the International Space Station to track small animals. So the animals with the battery, the power won't be generated from the tag. It'll almost be like your pet tags, you know, that you put in your pets. So it's like a microchip that go in the animals, and the International Space Station will send down a beam as it passes over and pick those animals up. So that's due to go on, I believe, in about 2016. So that's between the European and the Australian Space Agency. So that's going to be very exciting. We're going to be able to track huge amounts of much smaller animals, small birds and insects, which we can't at present.
I was just wondering about the longevity of the tracking devices. Like, I know it used to be only a couple of days, but it's obviously up to a year for those birds. But is it much longer yet?
Yeah, so the ones that we're putting in crocodiles, so this one I've got in my pocket, this is 10 years in that little device. Yeah, so there you go. That gives off a signal every minute for 10 years. The reliability of them is not so good, though. It's a little bit dodgy, but it's meant to be 10 years when we buy them. So yeah, the technology's gone better. But satellite, we're always, it's a huge, powerful signal to send that beam up there at the satellite. It takes a lot of power on the tag. So at the moment, the tags that you saw going on the crocodiles, they last for about 12 months. And that gives us 2 fixes a day for 12 months. So it's always a trade-off between how many times you want to sample the animal's movement and how long do you want the tag to last for, and how big's the animal so you can stick it on, and where can you attach it. And there's always those logistical considerations.
We'll just have one more question.
Could external trackers be solar powered at all? So like last indefinitely with solar power?
Yeah, so our tag's solar powered, yes. So for birds, that obviously works very well. But for most other animals, well, for like crocodiles, for example, it's no good because they're under the water. So for aerial species that are not living in the trees, it works very good. So Cyro have just brought out a new tag that they're using for flying foxes. And what that actually does is when the flying fox is hanging upside down during the day, the little solar panel flicks out and it recharges itself and it goes back. It does. And they can record like minute by minute during the night of the bats foraging, yeah.
Okay, I'm conscious of time. One more question. Quick question.
Actually, I have 2 questions. Like the first one is, firstly, you definitely know where they lay the eggs, the turtles, but do you know how long that they spend on giving birth to the turtle? Can you see that from your satellite?
They lay
data?
eggs.
Yeah, like how long that they spend, like how many minutes?
Marianne. There you go. They've been watching it. So there you go, 45 minutes. To lay 23 eggs or something.
Thank you. And also, I'm interested in tagging the juveniles of the turtle that you are tracking. Like
that was the juveniles we were tracking.
So you are interested tagging the parents and also the babies that they give birth
so
to.
the study I talked about was the juveniles, but we have been tracking the adults as well.
Okay,
we
thank
just
you very
haven't
much.
published that.
I'm conscious of time. I'd just like to take this opportunity to thank Hamish for the talk, but to invite Terry up to say a few words and then to follow that up with Professor Walker to close the evening. So Terry.
All the academics that I know that are men are tall. What is that? I want to encourage all the short men to persevere. I just really want to thank Professor Stephen Walker for the opportunity to have this event. If it wasn't so well supported by the university and the people involved, it wouldn't happen. So thank you sincerely. And thank you for letting us have Professor Craig Franklin, because he's really awesome at what he does, and we wouldn't want to be doing this work with just anybody. He's very fearless and very determined, and I really appreciate all his good work. And Hamish is amazing. He always inspires me, and I always learn something from him. And I think tonight I took a couple things away that I think were very profound. I think when he said, isn't it interesting that we cull crocodiles for reasons that may inconvenience us? And I especially thought, isn't it interesting, if a crocodile eats our pet, we want the crocodile to go away. We want to cull the crocodile. And I thought there is certainly one particular postman in Scotland that is very thankful that after he killed someone's pet, he was not culled. So it is an interesting reaction, and one that we don't have in situations that may be even more dangerous. I think that Hamish also made a really good point about safety with crocodiles, and it's pretty straightforward. You just don't swim with them. We've discovered through extensive research that crocodiles do not fly. They don't gallop along the road or come onto the football field and pull you off the field, and they don't live under your bed. You pretty much have to get in the water with them to have a problem with a crocodile, which is fascinating that we're the only ones with any ability or influence with crocs that seem to know that. So it's the same thing with the G20. There may be a lot of people that have left Brisbane, but I would still not encourage your children to play on the road, even if there are fewer cars. So even if there are fewer crocodiles, we probably shouldn't swim with them. So it kind of makes culling pointless, I think. I was also very inspired with the story of the Mary River turtle, because it really made me think about this argument about getting wildlife into the pet trade. And I think to myself, coming from America, I've never seen it work. But what I've heard over and over are stories, like we heard tonight from Hamish, about how the population of Mary River turtles may have been depleted by as much as 95% because of the pet trade. So I can give you countless examples of that. And if we think anyone is going to trade their cat or their dog for quolls exclusively, it won't ever happen. But if we start opening that Pandora's box, we have the potential of compromising a lot of wildlife. So I think it was very thought-provoking beyond the straightforward science of tracking animals and how important it is to learn about them. I was very inspired too by how many people showed up tonight with the interest, and I think, what would you have here, 100, 150 people maybe? I don't know how many people are here. 200 people maybe? How many are there? I just think we should probably track you and see what you're doing and how many you are. But I want to thank you all for coming and having that passion that Steve did. Steve was totally passionate about wildlife. And it was really good for television, and it was really annoying when you lived with him. So I'm a procrastinator because I'm normal. Like most people, I put things off as long as possible if I don't want to do them. Steve was the opposite. And what's fascinating to me is there's no word for that in the English language. What's the opposite of a procrastinator? We don't know, because there was only one of them. Steve was a do-it-now kind of guy, and I think everyone here shares that kind of desire to learn more and achieve more. So I'd like to encourage you all, if you are enthusiastic and passionate, to get involved. On Saturday, I think from 2:30, we've got conservation conversations. Say that 3 times. And we've got people just like Professor Craig Franklin, who are enthusiastic scientists, teaching us more about our wild world. And so I'd encourage you to come to Australia Zoo on Saturday and to get involved, because without your support and without people coming on board and helping by volunteering or financially supporting what we're doing, we couldn't continue it. So thank you very much, and I hope to see you all Saturday. Thank you. Thank you.
And finally, it's my turn to thank a few people. And first, I'd like to express my very warm thanks to Terry and the staff at Australia Zoo, and give you a little token of our appreciation of many years of collaboration and friendship. Thank you very much, Terry.
Thank you.
Yeah. And then, Hamish, I did have a question for you, but Craig sort of stopped before I got. I was curious how many years the tracker has been in your shirt pocket so far. You know, so. but seriously, I'd like to thank, and I think we'd all like to thank Hamish once again for a truly marvellous talk and on such an important issue of understanding our wild world and conservation of wildlife. And finally, I'd like to thank you all for your ongoing support for that area of science. So thank you all very much.